Well, good afternoon, everybody. Welcome to this afternoon's Faraday Institute Research Seminar. It's a pleasure to welcome you. My name is Keith Fox. I'm the director of the Faraday Institute. So it's a pleasure this afternoon to welcome Dr. Mirjam Schilling. She is presently working in the University of Oxford. She received a PhD in virology in Freiburg in Germany, and she's working as a virologist at present in Oxford. Her research focuses on the interplay of viruses and the innate immune system. At the same time as working as a postdoctoral worker on her virology, she's also studying part-time for a DPhil in science and religion, exploring the theological aspects of viruses. So Mirjam, it's a pleasure to welcome you this afternoon, and I think you're going to talk to us to the title of From Biology to Theology, How Viruses Challenge Our Perception of Good and Evil. Mirjam, over to you. Welcome.
Thank you very much for this kind introduction, and thank you everyone for joining and still being excited to talk about viruses. I think we're well into a pandemic, and I think after 1 year, I don't need to explain the types of suffering viruses can cause in much detail. The coronavirus has been around for slightly more than a year, and we've seen the impact it had on us, on our daily lives. But the coronavirus is not the only virus we see. We have seen other pandemics and epidemics the last several hundred years that we know of, and I think most people of my generation at least will recall Ebola, SARS, Zika virus outbreak, MERS coronavirus, the bird flu, and we've heard of the Spanish flu. But of course, epidemics and pandemics aren't the only times viruses come near us. There are millions and millions of people infected with virus currently. Especially chronically infected people are suffering long-term from viruses. Viruses can cause cancer. Viruses can also lead to disabilities. So there's lots of reason why we suffer from viruses. And as I've mentioned, we've all seen the areas in which we actually suffer from viruses. They're not just physical, they're also psychological, they're relational, and they're also spiritual. And depending on how your last year was, you'll most likely tick all or some of these boxes in relationship to the coronavirus as well. So the question is, how do we respond to this? And we've responded in political ways and also in very practical ways, by washing our hands, by keeping our distance. And even Martin Luther back in the days suggested that there are lots of practical ways in which we can fight a pandemic. In his case, that was the plague. And he suggests that God has created medicines and provided us with intelligence to guard and take good care of the body so that we can live in good health, and that we shouldn't tempt God by not doing any of these measures. So, but despite all of these political responses that we have and practical responses, I think there's still a different response that we need to have. And it's this inner response of how do we judge what's going on? What do we think is happening, and who is God in all of this? And over the recent couple of months, we have read books by leading theologians about the character of God and what they think the virus means. We've seen articles, for example, in the Church Times, but also more scientifically related articles that discuss whether or not viruses are evil. And even in the non-Christian circles, there are enough articles that discuss the meaning of viruses. So I'm quoting here one, which is called The Evil Virus Cell, How Students' Knowledge and Beliefs About Viruses, which is quite an interesting one, because they also think about what do people actually think a virus is, and not so much biologically, but the meaning behind things. So for the course of this talk, we will aim at thinking of the meaning behind viruses, but we look at it from a biologically driven perspective. So bear with me when we look at viruses and disease, and viruses and ecosystems, and viruses and where they come from. And we start with the biological perspective that I can give, and then we come to a close with more theological approaches, and I hope this is a help for everyone. So let's start simple. So what is a virus? At the first glance, it always reminds me a bit of the Death Star in Star Wars. It looks a bit scary, but also I hope you can appreciate the beauty of the virus particle we see here. This is a Zika virus particle, and dengue will look quite similar. So that's a virus family that doesn't have massive glycoproteins, but you can still see the symmetry and the beauty even without glycoproteins. But as you know, viruses do come in all different shapes and flavors and can look quite differently. Some with very prominent glycoproteins, some with less prominent ones. And as funny as the cartoons on the left-hand side look, you can see from the real images that they are not too far off. So viruses are not all the same, and viruses can be quite pretty. Viruses are also tiny. So here you can just see a depiction of a human red blood cell, and you can see the size of bacteria, like E. coli next to it. And you can see a very giant mimivirus in the middle. And that is one of the biggest viruses we know. But the average virus is much smaller. So you can see hepatitis B, which is a tiny virus, HIV, rhinovirus, all on the left-hand side of this image. And I think it's quite impressive that reagents that are that small have such a big impact on our health and disease outcomes. So when we look a bit closer at how viruses work, it's still intriguing because there's not a lot to it. So they all have a genome with instructions that explain how to build more virus particles. And they have these glycoproteins which they need to attach to a cell or surface. And everything in between is a lot of packaging. So they have proteins or lipids to package the system. And the main difference between all sorts of viruses, not only shape and the types of genomes they have, but also I call them tools. And these are basically a lot of accessory proteins that interact with the host machinery and help to interact with the cell and help to hijack it. And these can vary between different viruses. And there are some viruses that only have like less than 10 of them, and there are viruses that have more than 300. So the range in which a virus can interact with the cell varies a lot as well. So overall, a virus, as you can see, is not a lot of biology. And because it's not a lot of biology, it's also defined not as a non-living parasite. So it doesn't have a cell membrane, it doesn't have its own metabolism, and therefore it lacks cell-free production outside the host. To show this, you can imagine two jars of nutrients. In one, you put a bacterium, and the other one, a virus. The virus will just sit there, whereas the bacterium will happily divide, and you'll have a ton of bacteria in the very end. A virus cannot do any of that. It needs a host. So we call it life cycle, although you might think that life is a bit too optimistic for something that doesn't do a lot on its own. What a virus needs to do is find a host, enter the cell. They have different ways in which it can do that, depending on the type of glycoproteins or the type of virus we have. One of the main things it needs to do is evade our immune system as well, so that it can amplify its genome, which is usually taken up by the cell. Then there are components of new viral particles produced. These are assembled into new viruses, and they then leave the cell. And there are some viruses that happily leave the cell, and the cell is still happy and alive. And there are other viruses that exit the cell, and that causes cell death. So there are also differences in that respect. Right. We've seen what a virus does, but of course we're not helpless when it comes to virus infection. There's an arms race between a virus and our immune system. And this is an evolutionary arms race. So we've co-developed and we've both come up with systems and tools to overcome the other on the long run. But there's also a smaller arms race between each virus infection and each immune response. So what's the thing a virus can do? Except for the things I've already mentioned, the main tools a virus has is its fast adaptation. So viruses, when they replicate, usually don't have proofreading function. So that means when they copy their genome, there are random mutations that can occur. And because the virus is not alive and doesn't really care too much, there will be lots of virus particles that are no longer functioning, but there are also a lot of virus particles that either are the same and similarly effective or even gain some advantages. And it can do this quite quickly. Also, there are viruses that can build reassortments. So in the case of influenza virus, for example, if one virus infects a cell and another infects the same cell, because they have segmented genomes, they can basically exchange genome segments, and then you get a totally new virus out of that infection. We have viruses that can go into latency, which means they stick around for much longer. Herpes viruses are a case like that. And you know that once in a while they can reactivate, and then you get these little blisters or more severe infections. There are viruses that can integrate, like HIV, and so it's much harder or currently impossible to get rid of them at all. Viruses are really good at mimicry, so they can mimic part of the host, which helps them hide and fool our immune system. And they can also directly interfere with the host, so they can basically directly try to eliminate certain steps of our immune response, which makes it really hard to fight an infection. But as I said, we're not helpless. So there are certain tools that we have. And so it's important to know that our immune response happens in two phases. And the first one happens as quickly as a couple of minutes after infection, which is called innate immune response. This is rapid, as I've said, and non-specific because it's that rapid. So it's basically trying to figure out who's around, and if there's someone around that's not supposed to be there, it upregulates everything that could potentially help. And the second step, which happens much later in life, in the life cycle of the virus, and that is about probably seven days after infection, is the adaptive immune response, much slower but highly specific. And for that, we have B cells, T cells, production of antibodies. And that is also what we're aiming for when we vaccinate people, that our immune system learns how to very specifically fight a specific pathogen by building up the adaptive immune response. So we're not helpless, but the problem is that there's also a dark side to this fight. As I've mentioned, our immune system consists of two phases, and the way to communicate within the immune system is through cytokines. So here is an example which is called type 1 interferon. That's a cytokine and a messenger that is produced quite quickly after infection and leads to the induction of this very broad antiviral response only minutes to hours after infection. And the problem with that is that you really need to balance the response, because if your response is too low, you might protect your tissue from damage, but you might also have an increasing risk of pathogens surviving the attack. On the other hand, if you produce way too much cytokines, then you will definitely clear the pathogen. But with increasing amount of your cytokine, you also increase your risk for tissue damage. Because a lot of the symptoms that we feel when we are infected with the virus are actually not produced by the virus, but by our immune system. So you all know headache, tiredness, body ache, fever, runny nose, cough, sore throats, these things that you have just from a tiny rhinovirus, like during a common cold or the flu. And all of these symptoms are actually caused by immune responses and can't be the virus can't be made responsible for them directly. We also know and see currently, especially with COVID-19, that there are differences in which different people react to viruses. And there's some viruses where that doesn't play a big role, but for the coronavirus we currently have, it does make a big difference. And so we see there's a massive difference whether or not you're male or female, how old you are, if you have pre-existing conditions. And most of these differences can be linked to differences in our immune response. And not all of them are well defined yet. And this will probably take up years and years of research to see which differences are caused by what. But here are a few examples in which just the gender influences disease outcome for COVID-19. So the first of all is virus entry. There are differences between both genders in the amount of the receptor that the virus needs. So estrogens, which women have produced quite a lot of, downregulates the receptor. So there's slightly less chance for the virus to actually enter the cell. There are differences in virus sensing, which is the first step. So basically detecting if there's something wrong in the cell. There are differences in expression levels for these molecules that do the sensing. There are also differences in cytokine production, which means that all these innate immune response effector cells, like macrophages, NK cells, neutrophils, monocytes, in which they are basically guided to the place of infection. And there are also differences in adaptive immune responses, like B cell, T cell responses, antibodies. And there are more differences, and they're regulated sometimes by hormones, sometimes by age, and a quite complicated network of conditions. And there's a third example of why the immune response is also partaking in the way the disease develops. And this example comes from bats. So what we know about bats is super interesting. They have a wide range of very similar viruses as we have, but in contrast to us, they're not suffering from that. They don't develop disease. So these viruses can basically just spread through their population with no massive consequences. And the reason is that when you compare their immune system and our immune system, theirs have quite a few pathways that are dampened in contrast to ours. So in this table, you can see a few examples. In the left-hand column, you can see which pathways are dampened in bats, like TNF, NLRP3, STING. Doesn't matter if you don't recognize these pathways. But what's interesting is that a lot of the therapies we've developed over the last decades are actually not only targeting the virus, but also specifically targeting our immune response. And these mechanisms are targeting the same pathways that we see dampened in bats. So there is a dark side to our immune system. And I think the take-home message is that immune responses need to be carefully regulated within cells to not cause more damage. Right. So we've had a very anthropocentric view until now, and that's usually the view we take when we think about viruses, because when we think about viruses, we think about the one we are currently suffering from. But there's more. So instead of just thinking about one virus that spreads within our whole population, or even just our city or our family, we need to be aware of that we're actually part of a much bigger ecosystem, and that most viruses are not spreading only within one population, but they're spreading between different populations. And this has some consequences. So for example, for coronaviruses, we know that they probably all have their origin in bats. And since bats are not suffering from disease through viruses, the viruses are spreading in their population. And sooner or later, one of these viruses can jump to a different host. And if that virus then jumps to us or to a different host, then we are the ones suffering. But as you can see, it's not that easy. So to basically have a pandemic circulating in our population, a virus needs to overcome quite a few thresholds. So for example, in MERS coronavirus, the intermediate host was a camel. In SARS coronavirus 1, it was a civet cat. And for SARS-CoV-2, we're still looking for the intermediate host, might have been several, who knows, could be a pangolin. So, and that's not the only time and place where we see this jumping through different populations. We can also, for example, see this every year for influenza viruses, as you can see on the right-hand side. And there are lots of different species who can be infected with flu viruses. And they then jump from population to population. And this again changes the capabilities of the virus slightly and can cause a pandemic in humans. But given the amount of viruses we have on this planet, it's quite striking that we don't see this happen more often. And why does it not happen more often? I think one explanation is that viruses are so specific and they need to be so well adapted that each adaptation step is quite a big threshold. So they not only need to adapt to new host machinery because that's what they need to replicate, but they also need to adapt to new immune response where they hide from or that they can fight. And although this happens quite a lot, for us humans, we don't see too many pandemics, strikingly. So I've mentioned we have lots of viruses on this planet. Just to give you a few numbers, and I hope this does not make your next holiday near the sea in any way unpleasant. But when you just filter out what's in 1 milliliter of deep sea or coastal waters, you can see there's quite a lot of viruses in there. And how do we know that? And I think the main advantage we nowadays have is that we're no longer looking at one deceased patient and then try to figure out what's going on with the one virus. But the big advantage we have is that we can actually now sequence what's out there because of the technology we've developed over the last decades. And we get a much broader picture of what viruses are actually, what they actually are, where they are, and how they might interact with different ecosystems. And it's quite an impressive amount of number of viruses that we haven't known until now, and there's still quite an impressive amount of viruses that we haven't even seen yet. So where do we find viruses when we look at the bigger picture? Actually, we can find them pretty much everywhere, and we can also see how they interact in very different ways. So over the next couple of slides, we look at some of the ones that we have not mentioned so far. Scenario A, the antagonistic symbiosis, we always look at. We always see who's sick, and we always discuss how to fight that virus. But there are enough examples where they're mainly commensal, some of them are mutualistic, and other viruses even symbiotic. And we'll have a look at these now. So what do I mean with commensal? As I've said, we've started screening for viruses with sequencing technologies over the last years and years. And we found that there's quite a large proportion of viruses, even when we just look at the human virome, that are not causing any disease. We have viruses on our skin, we have viruses in our gut, and only a couple of viruses are actually causing or can be related to disease. And the same is true for all other species on this planet. It's not just mammals being infected, plants are similarly infected, bacteria are infected. Everyone can be infected by viruses, and it looks like a majority of them are not causing any disease at all. Mutualistic. What is mutualistic? In a mutualistic relationship, it actually means that one component is protecting the other. And there are now more and more examples, especially from plants, where a virus actually protects against either biotic or abiotic stress. So I've picked two examples to show you. For example, we have a wide range of white clover plants in our garden. And interestingly, they are less attractive to fungal nuts when infected with white clover mosaic virus or wild gourds. They're less attractive to beetles when infected with zucchini yellow mosaic virus. So there's something with that infection that protects them from other pathogens. And how about abiotic stress? So this is quite an interesting example about rice. You can see on the left-hand side an uninfected rice plant that wasn't watered for a long time. And on the right-hand side, one that was infected with the bromosaic virus before. And you can see the one infected can tolerate the drought much better than the one uninfected. So how about symbiogenic? I think that's even the more extreme example, because so far we've looked at viruses that acutely infect different species. But what if an infection becomes more chronic than you would imagine? So this is an example that we found when about 10, 20 years ago when we sequenced the human genome for the first time. And the interesting finding was that quite a big proportion of our genome consists of virus sequences. And so here you can see a bar chart for endogenous retrovirus sequences. So viruses that are belong to the same family like HIV does, for example. So they can integrate. And you can see that about 8 or maybe 9% of the human genome consists of these sequences. But there are more viruses that can integrate, and about half of our genome consists of sequences that could be coming from viruses originally, and that's quite a lot. So what does this do? And as I've mentioned, it's not just humans, it's basically all types of species. And here's a list of how integrated viruses can be used by their hosts and are actually a central part of these hosts. And we'll only look at the first example, the syncytin protein in mammals for a bit. But as you can see, there are more examples in more species with more different viruses having had a massive impact. So what's the syncytin? The syncytin is a protein that was originally found as a retrovirus surface protein, as you can see here encircled. And this protein was used by the virus to fuse with the cell so that the virus could actually enter. After this virus had integrated, and it integrated at different time points during evolution in different mammals. After integration, we are now using this protein, as you can see in the lower image here down there, for cell-cell fusion. And it's quite an important cell-cell fusion we're talking about, because this cell-cell fusion causes the formation of the syncytium, which is needed in the placenta. So all our pregnancies are impacted and supported by this protein. And I want to show a second example, and that's quite a busy slide. I talked about adaptive immune responses and how we need to produce antibodies to have a very specific response against pathogen. And here at the very bottom of this image, you can see how an antibody looks like. And there are certain variable regions. And the specificity of this antibody is produced by basically reshuffling of genetic sequences. And these are in the top panel here. And they need to be recombined so that they build these very specific regions. And the enzymes, RAC1 and RAC2, that mediate this recombination of genetic sequences to achieve this, these proteins are actually also derived from a recombinase of viral origin. So we are actively making use of a viral protein to fight viruses. And the third example of a very different kind I'm going to discuss here. So we don't only make use of viral proteins because they integrate it. When a virus integrates, it also integrates all the regulatory sequences. And as you've seen, viruses are tiny, and viruses are quite well organized to achieve what they achieve. So they have tons of regulatory sequences that come with them. And once the virus integrates, these regulatory sequences, of course, in the best case or in the worst case, depending on the outcome, can also help regulate our own gene products. And what this publication in 2016 suggested is that because many of these sequences coming from viruses originally could jump between different parts of our genome, because they could copy and paste or got excised and put somewhere else. This was actually an opportunity to regulate a whole set of genes all with the same regulatory factor. So imagine you have a remote control, and with just one click, you basically turn on your TV, you close your door, you open a window, and you start the microwave just with one click. And so this might have been an option where the cell used the regulatory sequence that was jumping to be able to regulate a whole set of genes at the same time. And the way they suggest this is when they look at all these cytokines that are immediately produced after virus infection. With one cytokine, you need to regulate more than 300 genes in the first instance to fight the virus. And so all of these 300 genes you could potentially regulate in a similar way if you had similar regulatory factors. And using the ones the viruses brought would have been one option to do this. Right, so that was a lot of detail. Let's come back to the big picture. But we've seen that viruses can be actually quite useful in different ways in interaction with their host. But what does this mean now for ecosystems? So when we look at a whole ecosystem, we can see that an ecosystem is made up of individual animals. These build a population. Different populations interact with each other, building a community. And the different communities, they build the ecosystem. So what happens if one individual is infected with a virus? What happens if that individual animal changes behavior or dies? You can imagine that one infection within one of these species can actually have a major impact on the numbers in that species, of predator-prey relationships, and that can change the whole interaction within one ecosystem. There's quite an interesting example that I think is very vivid, explains quite vividly of the dimensions of an impact such an infection can have. And it comes from the late 19th century when there was a pan-African epidemic, a rinderpest between different buffalo types. And what happened is that these buffaloes got diminished in numbers. And because there was less grazing pressure in that environment, there were more and more bushes and trees. But the fires that once in a while broke out could basically spread without any limitations. And so over time, in Tanzania, the area that they looked at, the ecosystem changed from a woodland state to a grassland state. The moment they introduced the vaccine and the buffaloes repopulated the area and the grazing pressure increased again, the ecosystem reverted back to having more trees and more bushes. And you can see that what I think is so interesting is that when we look at the world, we see how it currently is. And we don't estimate that a lot of what we see might also be because of certain numbers of animals and communities and how they interact, and that some of that might actually be caused by viruses as well. And there's another interesting example of how viruses are major players in our ecosystems. So here you can see a carbon cycle, and usually carbon would sink to the deeper oceans with animals that die. But because a lot of plankton is always infected by viruses, the plankton dies and doesn't sink to the lower areas of the ocean. And that means that CO2 is available in the upper spheres of the ocean and can be part of the atmosphere again. So there's a massive impact on the CO2 levels we have in our atmosphere through viruses. And the same thing again. So you have plankton that dies from a virus infection. And it's actually quite a lot of plankton that dies each day. I think it's about 20% of the overall plankton each day. And because it dies before it sinks through to the lower parts of the ocean, all the nutrients it had can be immediately taken up by other animals. Viruses impact the way algae bloom, and they massively impact the diversity, not only because they help supply nutrients, but also because of an evolutionary pressure. And they had on the development of all these different species. So there's quite a lot of impact that we don't even yet understand really, but we slowly get to see the more we sequence and the more we see viruses in their interactions with individual hosts. So I've just mentioned that viruses are also driver of evolution. Where do they come from? What do we know? And to be honest, we don't know a lot, but there are some theories, and I thought I'd present the main three to you, hoping that that would give you a glimpse of an idea of what we think happened. So theory one is the virus early hypothesis, which basically means in a time before the existence of cells, there was a pre-cellular stage at which some genomic elements and developed a way to replicate without the cellular environment, and that this was basically necessary for the development of cells as we know them today. The second hypothesis, called regression hypothesis, this hypothesis suggests that there was some cellular precursor called protocell, and that this cell then again lost its ability to do translation and replication and so on, and became a parasitic thing, which we now call virus. The third hypothesis, called escape gene hypothesis, and this hypothesis suggests that there once was a modern cell, and parts of the genome of this modern cell, they became selfish and autonomous and left the cell and are now one of these viruses. And I honestly think that it's probably all three of them in some aspect. So when you look at this image, I think there's quite a good chance that there were some replicative elements before cells developed. And I also think that with the development of cells, there's a good chance that these, by infecting these protocells, they picked up more things to build capsids and to build viral particles. And I think over time, the more cells developed and the more also the viruses developed. So I think there's a good chance that even slightly before the development of life through cells, but I think at the latest with the beginning of cellular entities, we also had viruses, and that up from then we co-evolved with the virus, so that there has always been this interplay between a cell and, as a host and the virus as a pathogen. And then we're back to the arms race in which both partners started to develop tools to overcome the other one. And just as a last reminder, that there's little we don't know. There's little information on this, but there's a lot we don't know. And but there are some early publications trying to figure out what the impact of this co-evolvement might have been. And there's one that I picked, and they suggest that potentially up to 30% of all the adaptive amino acid changes within the human proteome might actually be caused by viruses. Because as you can imagine, the virus is not only interacting with parts of our immune system, but it needs to enter the cell and is in touch and in connection with molecules there. Then it needs to transport its genome to the nucleus or somewhere else, and there are molecules it interacts there. And then there's cellular machinery that it needs to fit so that the cell produces more viral particles, and then the whole thing on the way out again. So there's a majority of proteins that are not involved in immune responses that are still in touch with the virus. And so there's a lot of interaction going on with a potential impact for development in both directions. Right, last topic, how many viruses are there? Do we know? Since I constantly say we don't know a lot yet. So there's one publication from 2013, and they've taken a lot of samples from a specific bat species. And they sequenced and found 58 different viruses. The interesting thing was that only 3 of them were known before. So there were 55 totally newly identified viruses. So when you then crunch some numbers and think that we know of more than 5,400 mammalian species, then this might mean there are more than 320,000 viruses that we don't even know of. Or if you think of the 62,000 vertebrate species, there might be more than 3 million viruses out there that we don't know of. And if you think of having more than 1.7 million known species, including plants and mushrooms and algae and whatever, then there might be more than 100 million viruses out there that we haven't even heard of. But it's even worse because this publication only looked at 9 different viral families, because you need to look for something that you're roughly familiar with. They did not include viruses of bacteria or archaea or other single-cell organisms. So this is most likely an underestimation of how many viruses we have not even described yet. To sum this up in a cartoon, lady asking, what is that? And the other one explaining, it's the virome and it's moving fast. Is that good or bad news? And the answer is yes. So I hope this does not confuse you or frustrate you, but the answer is yes. So let me summarize what we thought through today. What do we know about viruses and their biology? So we know that viruses are non-living obligatory intracellular parasites, which means they need a host. We encounter them on a regular basis. And it's not only the virus, but it's also our immune response that can determine the outcome of infection. We know that all species on Earth are affected by viruses, including bacteria. We know that viruses are involved in probably all processes in all ecosystems. We know that they have shaped the evolution of all species and that they can contribute genetic novelty. We have no clue how many viruses there are, but there is still a lot to discover. And so we're only beginning to understand their contributions to our lives, to our ecosystems, and how things work on this planet. So where does this leave us with our theology? I think one important aspect that we have to acknowledge is the individual suffering. And I think that's quite an important one and a very important pastoral task. But there's also a but, and I think we should not ignore the global contribution of viruses to the world we live in. And that includes our own existence and our own bodily functions. So I think this overall opens up a dialogue about the complexity of what we think good and evil is, complexity of answers to suffering. It opens up discussions about how we are connected with all living and non-living beings, and even our own human identity. So when we think about evil, to come back to the topic I've chosen for today, when we look through the lens of a virus, what do we learn? So I think there are about 4 things we might learn. First of all, we learn that natural evil is quite a subjective description because it depends on who you are and where you are, with whom you are, and what you rely on, whether or not a certain virus infection might be evil to you or might not be evil at all, or might even benefit you. I also think there's a contribution of our lifestyles and moral decisions, which we have not discussed at all, but there's a slight impact from the moral evil side of things. If you necessarily want to declare them as evil, I think it's very difficult to declare them as consequences of a human fall, since they've existed before humans existed in the first place. It's similarly difficult to declare them as consequences of a cosmic fall. Cosmic fall would be a nice scenario because it would mean that God is still good, and it would mean that, you know, biology fits theology. Viruses existed before humans. But the problem is that since they've contributed so much to how this planet currently looks like, that a lot of what we declare as beautiful and perfect in its ways was actually created through evil in this scenario. And I don't think this fits as well. So I think we might want to acknowledge that good and evil in itself is quite a complex topic, and that we might actually require a sort of compound theology, just as the one Christopher Southgate proposes. So there is definitely an impact of suffering to the creation of things and the evolution of things. And the challenge is to not only acknowledge that, but also live with that, while at the same time allowing a good God to still be good. And I think we need to see more components around just the suffering and just the evolution, like a better eschatology. We need to think more about the co-suffering of God, and in the very end, this might still leave us in a place where there's not a simple answer to things. So this is my conclusion for today. These are the things that I think we can learn. There are probably more, and I'm very much looking forward to discussing things with you and your questions. Thank you very much for listening.
Mirjam, thank you ever so much for that. That's a fascinating seminar, and there's already a number of questions that have come in, and can I encourage people to carry on adding questions in the list there, and I will see what I can do to forward them on to Mirjam. Some of them are theological, some of them you've touched on already in the talk, and might want to amplify a few of them as we go through. Maybe a technical question. Is it only viruses that cause diseases that have jumped from one species to another?
I don't necessarily think so. I think these are the ones we notice. So I think there's always this black box of things we don't notice, because as long as we don't notice, we don't start looking for things. But given the amount of viruses that are also in touch with either our guts or our skin, not so much in our blood, I think a lot of these viruses might have also come from other species at some point. I think the question is, are we discussing the last 5 years, or are we discussing the last 500,000 years? Given the evolutionary trajectory, at which point do we think a thing crosses a species? So I don't necessarily think they need to cause diseases, although I think there's this paradigm that thinks, once you jump a species, you're not well adapted to your host, which means that the likelihood that you cause some sort of incident is much higher than that you go smoothly along and no one notices. But you might adapt quickly as a virus, and then you're part of a new species host interaction. So I think it depends a bit on the time point we're discussing here.
Yeah, okay, thank you very much. You've given some examples of evolutionary time where viruses have benefited us. Are there any viruses that infect us now that are a benefit?
That's a very good question. Yeah, that's a really good question. So I don't think we know a lot about that, but I think there are some hints that actually might be the case. So and not only with the viruses that so there are viruses infecting our gut bacteria, and we all know by now that the gut bacteria are quite important when it comes to our own immune response and how we deal with infections. And I think a lot of that will also be navigated by the viruses, because they will determine which gut bacterium reaches a population size and which doesn't. So I think that's one example. Another one I can quote, because there was a study looking at that. So we have lots of viruses that are sticking with us because they're either integrating or go into latency, like herpes viruses. And there was a very interesting study that said that people who are CMV positive, so CMV is one of the herpes viruses that roughly 30 to 40% in the UK will probably have. And these people are actually in an advantaged situation when it comes to flu infections. So they have much better immune response and can fight off flu much better. Whereas, like, I know I'm CMV negative, so I'm not making use of that. This is not benefiting me. So that's one example where we know there's a beneficial interaction.
Thank you. Very early on, you asked the question whether viruses are living, and they're not living in themselves. They infect cells, and yet we can't resist the temptation to use anthropomorphisms. I think you described them as mimicry, which is a sort of, are they living? Can we, is there a way in which we would say that a virus is alive?
That's a wild discussion, and that's one that I think is really interesting. So first of all, per definition, currently they're not alive because they don't have their own metabolism. But there's a but. So about 10 years ago, they discovered these massive viruses, and they've shown the Mimi virus on one of my first slides. And in the first instance, they were mistaken for bacteria just because of the sheer size of them, and you could see them with a normal microscope and stuff. So that was super interesting. But also, like, the bigger a virus is, the more tools it can bring. So the more and more independent it gets from a cellular system. And so that actually quite triggered the discussion again of whether or not viruses are actually could be considered as alive, because there are also other pathogens who need one or two things from a host to actually be effectively replicating a lot. So what that would mean is, like, where do we set the limit between, that bacterium that still needs something from my host cell is living, but that virus that only needs the same amount of things from the same host cell is no longer alive. So I think there's a discussion going on about how difficult it is to make a unifying definition of life that is valid for all species and all things we see on the planet, and has a strict threshold of setting aside that's no longer alive, that's a stone, and that is alive because there's a virus. So I think there's a very interesting discussion that will continue for a while, I think, and might end up in a very philosophical, because what actually do we think recognize as alive just because it's alive on this planet. We're currently looking for life somewhere else, like on Mars. Would we recognize that as living? So I think we're also in a position where we know what we think, what we're looking for, but life might be a bigger topic. We need to think in less or in different ways. Yeah, we might be very limited.
Thank you. I mean, a question that begins to ask questions a bit more theologically. Should we distinguish between a virus and the disease that it leads to, as we're talking about evil and suffering? Can we say that one is good and the other is evil, or how do we distinguish between those?
Yeah, I don't have a clear answer for that personally, because I think it's difficult. Because it also includes our discussion of what do we think is harmful. Are we thinking short term? Are we thinking long term? And there's this interesting Taoist story about this farmer who had a son, and then the son fell off his horse, and everyone complained and said, "Oh, we're so sorry for broke his leg, and that's really sad." And then the next day there was a war, and all the sons of the other farmers had to go into war, except for that son because he had a broken leg. And everyone was like, "You're so lucky, happy son, broken leg, much better than going to war." And so I think it's this discussion of what do we think is beneficial, and is it my own interpretation? And do we need to not only think about, is the virus itself not evil but the disease, and then put a threshold there, or is it more complex when we think of beneficial things? So something that could harm me now could be beneficial later on. So like the CMV I mentioned might be beneficial if you catch a flu infection, but in the first instance, it's still infection with the herpes virus. And if you're pregnant at the time point of infection, that could actually lead to disabilities for the newborn. So there are things that are more complex. And I think I don't like putting things in categories nowadays, because I like, whenever I have a category, I think it turns out to be more complicated than I thought. So, or we put things in categories and know that, in a year's time, we need to think of them in a different way and start all over again. So I don't know, it's not a good answer to this, but I think, yeah, things are more complex.
Thank you. So a questioner asks if you could expand on your views of how our knowledge about viruses affects our views on the nature of good and evil. You touched on that at the end. Do you want to say a little bit more about that?
Yeah. So I think one thing I think is we need to distinguish between moral and natural evil. And I think the moral evil is probably the one biblical topics touch on more often. And I think that's also the one probably more important for my relationship with God and with other human beings. So, and it depends a bit on what you think is important, but I think that a lot, when we think of eternal life, is going to be manifested in my relationship with God. So what happens on this planet will indirectly affect that, but it's not going to be as important. So I think that there's these categories, moral evil and natural evil, that have different priorities, I think, in one place, at least when it comes to my relationship with God and my eternal well-being. But from a pastoral point of view, of course, these are of similar priority. So you can't say that this prioritization is always the same. So when you look pastorally, one person suffering from a virus infection or from a disability because of a virus infection, or because someone else died from a virus infection, that pastorally is still the same issue, and that needs to be recognized as suffering and evil, and might also need recognition that there is that it needs reconciliation in the relationship to the divine as well. So it's not just about emotional well-being, but there's more to that. And suddenly moral evil and natural evil become like the same in terms of priority. When it becomes important to how do I personally act in this world, then there are two options, because I can either say, well, natural evil I'm no longer concerned with because it's not really affecting my relationship with God. But that's also thought through too short, because like if my moral sin affects the natural world, which it constantly does, not just with viruses, like do I pursue animal markets? Do I cut off half the trees in the forest, making all animals come closer to the city, which allows jumping from virus to us more quickly? So there's all these different implications. So I think it depends a bit on the different situations we look at. So I think it's hard to discuss good and evil as a general topic, which we currently do a lot. And I think that is still important to once in a while discuss. But I think there are all these subgroups of good and evil, and why are we discussing them? So I think a purposefully discussion of aspects of good and evil might be more helpful than as interesting as a philosophical debate is. But I think the philosophical debate will come to its limitations quite quickly.
Thank you. Thank you. The question about the current pandemic and the future, is the current pandemic, was it inevitable? And will there inevitably be other ones? And if so, are there any things that we can do to prevent them?
Inevitably. I think it was inevitably in that sense that sooner or later we did expect the coronavirus to jump again, because we've seen them jump before. They're quite common viruses. We have them circulating in our population, which means we know that they can make it in our population, and they find ways to adapt. So in that sense, yes, inevitably. I think what very difficult is, and what we're still suffering from, and that's why it's global and that's why it's so hard to fight, is that it's quite, let's say, an unlucky combination of things within that one virus. So spreading before we develop symptoms makes it so hard to isolate infected people, because they basically infect 5 people before they even know they got the virus. And so we have this massive pandemic going on, and we're all sitting in home offices now for quite some time, because there's no other way to fight this. I think in that sense, I don't think it was inevitably, but it was an unlucky combination in that sense that, if it's, and plus it's spreading easily, you know, it's in the upper respiratory tract, when can replicate there quite well. So it's a combination that's not good for us. And so I think in that sense, we're quite unlucky. I think we can expect more pandemics to, you know, show up the next centuries. That's not a surprise. But not all of them might turn out with that unlucky combination for us. So they might be easier to fight depending on what they are. And I think what can we do to prevent them is a very good question. So I think there's lots of things we already do. I mean, there's lots of monitoring, and usually when there's something unusual going on, it's reported quite quickly, and that did happen the last time as well. I mean, could have always, you can always report things quicker. So there's this blame game going on. We should have, you know, they should have told us earlier, that we could have done something. But honestly, would we have done something? Because a year ago we still thought, you know, that's a virus that is spreading in China, but you know, it'll never get to us. Yet here we are. So I think, I think the blame game is not helping. I think keeping monitors, keeping open eyes, doing lots of basic research to understand viruses better is always a good thing, because that's the only way why we could develop these vaccines so quickly. And you all know that's quite an unusual time frame we have managed with vaccines that is super helpful and will be the way to fight the virus in the very end. That's the thing we can do. There's this general eco-theology that we could apply to how are we actually treating other species? Are we how are we limiting the space where they live? Because the closer they come and live with us, the more likely it is that a virus jumps and adapts. So if we cut down massive forest areas, if we destroy their areas of life, we will have problems with that. The way, how do we, you know, treat animals in terms of food industry? There are all these issues that we need to discuss long term.
Thank you very much. I think we'll have to finish our questions there. There's lots more that I could give you, but I think we've run out of time. So can I thank you for giving us an excellent seminar this afternoon. I've learned plenty, and I come as a biochemist to this. You've informed us, entertained us, and made us think. And thank you ever so much for being with us this afternoon. Can I say, for those of you who want to pursue the topic further, the Faraday Institute are giving a lecture as part of the Cambridge Festival Bob White and Roger Abbott. That'll be Friday the 26th of March at 2 o'clock online. The details are on the Faraday Institute website. You might look at that and then register for attendance at that as well. But in closing, Mirjam, thank you ever so much for being with us this afternoon. And thank you, everybody, for putting your questions, for being with us. This is the last seminar of this term. I'll be back with you again at the start of next term, and look out for details in our announcements of that. So with that, I will close, and thank you for being with us today. Bye-bye.
Bye.